discovery of Gregor Mendel, his experiments with pea plants revealed two key principles of inheritance that apply to all living organisms: the law of segregation and the law of independent assortment. So the law of segregation states that during gamete formation, each parent actually contributes only one allele for each trait. This ensures that offspring inherit one allele from each parent. Where the law of independent assortment explains that traits are inherited independently of one another, as long as the genes for those traits are on different chromosomes. So these principles actually allow scientists to use Punnett squares to predict the genotypes, or the genetic makeup, and phenotypes, or physical traits, of offspring, which helps us understand patterns of inheritance in plants, animals, and humans. We also have the principle of dominance. So, like we talked about in our last lesson, our dominant trait is always going to show up if it is present. So we're going to talk about each one of these laws of inheritance. So, the law of segregation. Each parent contributes one allele for a trait during reproduction. Where your law of independent assortment, traits are inherited separately. So your fur color doesn't affect ear shape inheritance. All blondes are not tall. Everybody who has brown eyes do not necessarily have brown hair, things like that. So just because you have one trait doesn't necessarily mean that they're always going to have the other. So they're inherited separately. The principle of dominance is the dominant allele is expressed. So while the recessive allele is masked and only appears if two recessive alleles are inherited. So for the example to the side, this heterozygous genotype, it has a dominant allele. See that uppercase T? So this uppercase T is going to express that dominant trait. Even though it has a recessive trait, this dominant one is always going to be expressed because it is a dominant trait. So think of some really, really big person and a really, really small person. If they were to play tug of rope, then the big person is always going to win because they are stronger, they're bigger, and they can pull that little person, maybe it's a child, over, right? Where your recessive allele is not ever going to win. It's always going to be masked. So let's look at some types of dominance. You have complete dominance. So this is the dominant allele completely masks the recessive allele in the phenotype. So if you have this uppercase T represents tall and this lowercase represents short, then this person's going to be tall because they have that dominant gene. An incomplete dominance is the phenotype is a blend of the two alleles because neither is completely dominant. Your example here, if you have a red rose and it breeds with a white rose, you're going to get a pink. So neither of them are completely dominant, so it's not going to be just red or just white. It's going to be a mix of both of them. Codominance. This means both alleles are fully expressed in the phenotype without blending. So if you have a white cow and a black cow and this codominant trait, you're going to get a black and a white cow because both are going to be expressed. A sex-linked type of dominance would be a dominant allele located on the X chromosome, affects traits differently depending on biological sex. So if you have a dominant allele that is located on the X chromosome, you're going to have it expressed, especially when it's an XY. If it's female and it's XX, then you might have it on one side and not on the other, and it won't be expressed. We're going to talk about each one of these in more depth in upcoming lessons, so don't worry about it too much right now. Just know that there are different types. And the last thing I need you to know for sure for types of dominance is also the generation levels. So your P generation would be your parental generation. This is the original pair of organisms that are expressed or crossed in a genetic experiment. So think of your P1 would be parents. Your next level would be your F1 generation. So your F1 generation is the offspring of the P generation. So your P generation is your parents. The F1 would be the kids. And then finally, the F2 generation would be a cross between two members of the F1 generation. So that could be grandkids or kids of the F1 generation. So let's look at your actual method of calculating the chances of inheriting a specific trait. So this is your Punnett square. So your Punnett square is used in order to show the chances of inheriting these specific traits. So let's look at this a little closer. The step one of using this Punnett square would be identify the parent genotypes. So you're going to write down the genotypes of both parents, the combination of alleles they have for the trait. So here's an example. A black bull is breeding with a black cow, and what is the offspring going to look like? So if we go over here, we can see that a black bull can be either BB or heterozygous, uppercase, lowercase b. Red is homozygous recessive for lowercase b. So we know that with a black cow and a black bull, we're going to have either capital B's or heterozygous. So we're going to have homozygous dominant or heterozygous genotype. So we're going to look at either one of these for our parent genotypes. So step 2, you're going to set up the Punnett square. You're going to draw a 2x2 grid, so there's 4 squares total. So something like this. And once you've got that, you're going to write the alleles from one parent along the top. So here's the black bull. We're going to write one allele to the left and one allele to the right. Then you're going to write the alleles from the other parent along the side. So one allele beside each row. And we're going to do our heterozygous alleles on this side and our homozygous dominant on this side. I always get the question, does it matter where you put them? So could we put the black bull on this side and put the black cow on top? Oh, why yes, you can. It really doesn't matter which side they are. They're going to get the exact same results. So after you've gotten that, you're going to find the F1 generation. So you can see that your F1 is here and here. So your P1 would be the parents of your black bull and your black cow. So step 3, you're going to fill in the squares. So stay with me because we're going to combine the alleles from the top and the alleles from the side. So we're going to bring one over and one down. So we've got uppercase and uppercase. You can see that we bring this allele down, which is this one, and this one over. So here's another question. Does it matter which one comes down and which one comes over? Not necessarily, because they're both going to end up in this box. What matters is that you have the dominant trait first. So this one has 2 dominant traits, but if we look, this one's going to have 2 dominant traits as well. If we go down to this bottom one, we're going to bring the uppercase down first, and the lowercase is always behind. So make sure you remember that. Your dominant trait is always going to be first, and your recessive trait is going to be last. So we do it again for the fourth square. Bring it over. So we have two squares that are homozygous dominant and two squares that are heterozygous. So these are your P1 generations, is these offspring in the boxes. So you're going to look at the genotypes in the Punnett square and you're going to count how many boxes have each genotype. So, for example, your B, or your homozygous dominant will have 2. Your heterozygous will have 2. And your homozygous recessive has 0. So your genotype ratio is your homozygous dominant, right here, 2, heterozygous, 2, and then your homozygous recessive, 0. So it's always dominant, heterozygous, homozygous recessive. In order to find your percentage, you're going to divide the number of squares for each genotype by 100. So 100 divided by 4, you're going to get 25. So 100 divided by 4 is 25. And then you're going to multiply it by this number. So 2 times 25 equals 50%. So you can see half of them are homozygous dominant. Again, 2 times 25 is 50%. Half of them are heterozygous. 0 by 25 is 0%. None of them at all are homozygous recessive. So you can see there, 50% and 50%. You're going to determine the phenotype ratio next. So you can see that your genotype is 2 and 2 and 0. We're looking at phenotype. So what would this square be? Would it be black or would the cow be red? The same with this one. Would it be black or red? How about these two squares? Would they be black or red? So these two would be black, and so would these. Because remember, you've got a dominant trait, and black is dominant. So you're going to have all 4 offspring would be black. Would it even be possible to have a red offspring? No, because you only have 1 recessive allele. If you had a recessive allele here, if you would bring it down and bring it down, you'd have 2 lowercase b's, or a homozygous recessive, and that would be red. So 100% is black.